this was full on my laptop, so that's not good. So you guys hear these commercials, right? Lysol kills 99% of bacteria. I mean, you will realize after this unit that 99% is not really a great number because there's billions of bacteria. And then even when a product kills 99%, it just simply means that, if you think about 10 to the power 12 bacteria, 99% of that is not really a lot. And there's still tons of bacteria that's left around. So when we talk about bacteria, we talk in billions, literally 1 ml of water can hold millions of bacteria. So bacteria are everywhere. And then we'll talk about generation time, and then we're going to be talking about the serial dilution mechanisms and how we can dilute the bacteria to actually come up with the most probable number, how many cells that might be present. And so there's different type of bacterial cell division. So there is binary fission. We might have, that's the most popular, the most well-studied type of cell division is the binary fission, which means the bacteria simply divides in equal size from one cell to two daughter cells to roughly same size cells. And then there is asymmetric division or buddying, where usually there is a polar division. Only one pole of the cell elongates and then divides. So the newly formed daughter cell is usually smaller than the parent cell. So those are called asymmetrical division. So binary fission is highly symmetrical, so that there's a division of the cell right in the middle into two equal sized daughter cells. But asymmetrical division is when the daughter cell is not the same size. And then there's budding. Obviously, you might have heard about it. Yeast, Saccharomyces cerevisiae, it produces a bud. So the bud simply just separates from the parent cell and then grows into a new adult organism. And then there's fragmentation. Cyanobacteria, the only photosynthetic bacteria we know of, undergoes division through fragmentation. So normally the nucleus divides, not nucleus, the nucleolus divides into multiple copies, and then usually there is a formation of the cell wall around each nucleoid. I might have said nucleolus again, sorry, nucleoid. Around each nucleoid, and then which later separates. So almost like the cytoplasm itself or the parent cell itself fragments into different parts, each containing its own nucleoid. So let's talk about binary fission. So as I mentioned, binary fission is the most common, most well-studied mode of cell division. E. coli probably has over thousands of articles on cell division and what are the different proteins involved in cell division in binary fission. So the cell division in bacteria is tightly regulated. So in case of human, we say our generation time is about 25 years, roughly, approximately. So humans, we reproduce by the time we reach puberty, but in our 20s. But in bacteria, it's not like that. The generation time is widely diverse. Some bacteria like E. coli, for example, divides every 20 to 30 minutes. And then the same bacteria, when you change certain environmental condition, it could live up to hundreds of years or even thousands without showing any division. It's pretty remarkable how they survive in the environment because it totally depends on the environmental condition, type of the nutrients available, temperature, moisture, a lot of different conditions. Ideally, we say the generation time is 20 minutes. That's when you put E. coli in a nutritionally dense, nutritionally rich media, 37 degrees Celsius, yes, it divides at 20 minutes. You take the same bacteria out, put it in a media with nutritional deficiency, like loss of, lack of certain amino acids or sugars, then suddenly the cell division stops. So it totally depends on the environmental conditions. And then obviously the binary fission involves the most important step, which is the replication of the DNA. As I mentioned, the DNA in case of prokaryotes is circular. So it's covalently linked circle of long strands of highly compact supercoiled DNA. And the division always starts at one point on the DNA, and that's called the origin of replication. And it's always and only starts at that specific point. So it's got a specific set of nucleotide sequences, and the DNA polymerase always binds to that spot on the DNA. And the origin of replication is also called ORIC, O-R-I-C. And that segment of the nucleotide sequence is always connected or linked to the inner membrane. And that's where the the DNA polymerase binds. And then eventually there is a recruitment of a number of different proteins. And then that's where the replication starts. Now, if we're talking about a rod-shaped bacteria, a life cycle of bacteria involves, first, as soon as you put a bacteria in a nutritionally dense media, the cell prepares itself for division. So it accumulates a bunch of amino acids, sugars from the environment, and then there's lots of molecular changes happening inside. It's essentially growing in size and prepares for the DNA replication and prepares for the binary fission, basically. So there is a stage where the cell, if it's rod-shaped, essentially becomes longer. And once it reaches a certain size, then you will start to see like a furrow right in the middle where all the different binary fission proteins, we call it the Z ring, composed of over 20 different proteins, they accumulate right in the middle. And then that's where the constriction happens. But before that, there's two different complex mechanisms that we talk about. One is the formation of a divisome complex, which is the second stage. The first stage is the formation of the elongosome. Elongosome contains roughly the same number of proteins. I don't expect you to remember or memorize any of these proteins because, for God's sake, I can't remember them myself. The most important class of proteins are the penicillin binding proteins. So those are the PBPA series of proteins. They're called penicillin binding proteins because that's where the antibiotic penicillin or the beta-lactam class of antibiotics bind and stop the production of peptidoglycan. I kind of mentioned that in the previous classes. That's how it targets prokaryotic cells because that's the characteristic feature of prokaryotes is the presence of peptidoglycan. So the divisome, the elongosome complex, along with all these proteins and penicillin binding proteins. So the penicillin binding proteins are the ones that recruit the addition of peptidoglycan precursors to the cell wall elongation. So when the cell becomes elongated or grows in size, essentially it's adding new peptidoglycan components so that maintains the same shape and then the rod essentially becomes longer. It's growing in size. So one of the main steps is addition of peptidoglycan precursors to the right and which adds to the length of the cell, that's how the cell essentially becomes longer, it grows in size. And then at some point in time, the DNA replication starts. And then once the replication has progressed to a certain length, essentially when you have almost two separate nucleoids, the elongosome basically just disperses, and then there is the second complex comes into picture, which is the divisome complex. I've talked a lot. Any questions up until now before I jump into the divisome complex? Like I said, you don't have to remember any of the specific proteins. I won't ask any of the about the name of the protein exam, but then you just have to know the general mechanism. When does elongosome comes into picture, and then when the divisome comes into picture, and what are their roles? Any questions so far? Anything confusing? No? So yeah, lots of penicillin binding proteins, and some of these proteins, like MREB, a series of proteins that are actin-like proteins. Essentially, they're providing kind of like a structural scaffold. Prokaryotes don't have like an active cytoskeleton, but then there are proteins like MREB. They are like actin-like proteins, for example. Like eukaryotic cells have actin and tubulin that forms like a matrix, but the MREB series of protein that are acting like, in the sense that they have a similar biochemistry, and then they produce, they offer like a scaffold, like a supporting structure for the elongosome, where the proteins come and go, the addition of peptidoglycan precursors, and the cell is growing. So the divisome comes into picture after the nucleoid has completely or almost completely has divided. And again, the divisome complex contains over 20 different proteins. And then the main one that's really been well studied is the FITZ protein. So there's FITZ, which forms the main scaffold. I'll show you like a couple of fluorescent images of FITZ GFP, because I did quite a bit of work on FITZ GFP. So the FITZ forms what is called a septum right in the middle. The septum is essentially a series of all these different proteins that form like a ring right in the middle, and that's where the constriction happens. So FITZ allows constriction of the cytoplasmic membrane, whereas the elongosome does not have any of the FITZ proteins. So the elongosome contains just the MREB and the penicillin binding proteins that helps in the growing of the cells, increasing in the length. But the divisome allows the constriction of the cytoplasmic membrane, because it comes into picture later after the nucleoid has almost fully has divided. So the FITZ, along with a bunch of other protein like FITZ-A, and then there's ZAP-A, and like almost 20 different proteins, they form a complex which helps in the constriction of the cytoplasmic membrane. And then there is so the combination of all these 20 different proteins forms the Z ring, which is right in the middle. The cytoplasmic membrane constricts on both sides. And then there is, again, addition of peptidoglycan precursors, because the penicillin binding proteins also come into picture almost in the, as the cytoplasmic ring is constricting, the cytoplasmic membrane is constricting. There's also addition of new precursors, the cytoplasmic precursors, and there is addition of peptidoglycan precursors right in the middle, which helps in the constriction. And then the cell eventually divides into two daughter cells. Again, there's lots of different proteins. You don't have to memorize any of them. But is that clear so far? Yeah. I took a lot of pictures of cells that were labeled with the FITZ GFP. So these cells are alive in the sense that the tag, the cell itself produces FITZ with a GFP in it. So as the cell is dividing in live, you can see the signal coming from the septum. So in the top row, so the DNA is labeled with the DAPI. It's a fluorescent dye that binds to the nucleoid. And then the GFP, the ring right in the middle. So the very first image has a combination. It's got both signals coming, so you can't really see the Z ring. But in the middle, when you separate the two signals coming from GFP and DAPI, so confocal microscope allows you to do that. You can do something called linear unmixing that just separates the two signals, because the signals are coming at different wavelengths. So the DAPI is blue, GFP is green. You can easily separate them, and you can see. So there's the formation of the Z ring right in the middle, and then the DNA is divided. You can see them two separate. So the division is really interesting. Sometimes even before the cell has completed its division, the daughter cell is already preparing for the next stage of division. And this happens especially when the media is really rich and there's optimal condition. All the cell is doing is just basically eating and dividing. Even before the first cell has finished dividing, the daughter cell is already preparing. And I was able to see, so there is the two daughter cell with the two Z rings, and sometimes even before the constitution is finished, the daughter cell is already accumulation of the FITZ right in the middle of the daughter cell. So it's like a really fast process. Yeah, at the bottom panel, there's lots going on because it's treated with 2,4-D. But it's kind of stretched. At that point, it's not really normal anymore. So what I was seeing with 2,4-D was there's like multiple bands, multiple Z rings, and the cell was not really doing the constriction. So the daughter cells were not separating. There's lots of partially formed Z rings in those long filaments images I had showed. It couldn't divide. So exactly, yeah. So there's lots of partially formed Z rings. Because the cell is undergoing stress, there's lots going on. But yeah, it's similar to like what you're saying. So I was seeing cells where there's like a Z ring in the daughter cell, and then as it's separating, there's more Z ring. So yeah, exactly. Yeah, it's similar to that, but then I can't really say that's what happens because it's treated with 2,4-D. Yeah. Previous one, yeah. Yeah, I don't expect you to remember that. Sorry? Yeah, no, you don't have to memorize it. I just wanted to give you a picture of what the protein complex looks like. There have been articles, there's lots of articles that there are articles that simply focus on one protein and on its role and what kind of experiments they've done. It's pretty intense and it's really in depth, and I don't think you need to know that at this point. I mean, if you're curious, obviously go ahead and read all those papers, but I don't expect you to remember those for the class. Okay, any other question? Okay, so let's talk about generation time. Generation time, as simply the term defines, it's just a time between two, or I should say the time that takes E. coli to become 2, basically. So the time between time it takes for the cell from becoming the doubling time, basically, is the generation time. And then, like I mentioned, bacteria, the generation time could vary widely depending on the environmental condition. But then for convenience in microbiology, we kind of define a generation time as, considering that the conditions are optimal for division. So we say E. coli generation time is about 20 to 30 minutes, but then obviously that's under optimal condition. And when you change the condition, the generation time obviously changes. And apparently there are bacteria that live deep down deep below Earth's surface that divide every 1,000 years. It's pretty remarkable because clearly the environmental conditions are not suitable. So the bacteria essentially is in like almost like a dormant state. The metabolism is in such a low speed that they don't divide for hundreds or thousands of years. They're essentially waiting for an optimal condition to arrive. And then again, I don't know if you guys heard about how we make glycerol stocks in the lab. So have you heard about how that's done? So if you have a strain of bacteria, if you're simply just culturing and reculturing in the lab using the same bacteria without preserving it, the bacteria undergoes a tons of mutation, and then by the 10th generation, it's no longer looks like physiologically or genetically it's same as the strain that you started with, because they mutate so much. And so what we do in microbiology is you inoculate it a few times, and then you basically want to make a glycerol stock. You want to keep using the same bacteria because it just changes so much. And the glycerol stock is, glycerol is a sugar alcohol, so it's a preservative. So we make like 40% dilution of glycerol, mix it with the culture, put it in the freezer at -80, and then it just stays there for infinite amount of time. And then you take it out, you thaw it, and then you streak it, and then you get a fresh culture. And the culture is identical to the one that you froze. As opposed to if you just didn't freeze it, you just keep re-inoculating, keep growing it. I mean, it'll grow, it'll multiply, you'll get a culture, but then it just changes a lot. So some bacteria like Clostridium, for example, have a generation time of 10 minutes, whereas others like a Mycobacterium tuberculosis, up to 12 to 16 hours. And that again depends on the genetic makeup. So Mycobacterium tuberculosis is a human pathogen. And I suspect, I don't know if this number is taken from like when the bacterium is inside humans, that it's 12 to 16 hours when it's outside. I'm not so sure, but then I guess again the point is a lot of different conditions matter when we're talking about the generation time. And then you will, I don't know if you'll be doing it in the lab, but then if you know the generation time, you can kind of calculate how many cells you will have using a simple equation. For example, if you consider E. coli, let's say you consider that it has a generation time of 30 minutes, and then by the end of 48 hours, how many cells you're going to have. But you can do that by simply using that equation. Fairly straightforward. So because it's not, you say doubling time, it's exponential increase in logarithmic increase in the number of cells. It's not just like double. So 2 becomes 4, and then 4 becomes 16. So it's 2 to the power. So I think you will know what I mean. It's a simple, fairly simple math. Any questions about generation time? Okay, so let's talk about bacterial growth curve. So again, for convenience, we consider the sigmoidal growth curve here based on the consideration that we're talking about bacteria in an optimal growth environment. As soon as you inoculate a certain number of cells in a fresh media, as I mentioned, there's going to be a lot of metabolic changes happening. Essentially, the cell is just sensing the new environment. It's just preparing its metabolic machinery for the uptake of the sugars and amino acids. It's just preparing itself for division. And that phase is called the lag phase. So at lag phase, for E. coli, it's usually the first hour or two. As soon as you inoculate, you put your culture in the media, in the incubator at 37 degrees Celsius. It takes some time for the bacteria to get used to the new environmental conditions, prepare itself to divide. And that phase is called the lag phase. And usually the number of cells don't increase at that point. So the curve remains flat. And then as soon as the cell enters the log phase, which is the second stage where now the cell is just ready, just all it's doing is dividing. There's an exponential increase in the number of cells. As I mentioned that formula before, it's essentially exponentially increasing. The doubling time is just so fast. The optical density, we'll learn that what that is later on, just a way of kind of measuring the turbidity. Because as the cells are dividing, the media becomes turbid. So at log phase, there's an exponential increase in the number of cells. And that's because the bacteria is happy, lots of new nutrients in the environment. So that phase is called the log phase, or the exponential phase, where the number of living cells, bacterial cells, is increasing exponentially. And then there's the stationary phase. As the name itself says, there's bacterial cells. The overall number of cells don't really change as much because if you don't supply any more nutrients in the media, then the media is essentially running out of vital nutrients. So some of the cells start to die. Again, depends on the competition. So there's lots of research just done on what happens to cells at stationary phase. There's lots of genetic changes happening. So the bacteria that are able to somehow absorb the last bit of nutrients, they still keep dividing. Whereas the bacteria that are kind of weaker, they're maybe not so good at competition, start to die off. So overall, the number of cells don't change so much in stationary phase. That's why the curve remains flat. And that for E. coli is usually, the end of log phase is about 6 to 8 hours. And then after 6 to 8 hours, the cells enter into stationary phase, which is probably like another 5 to 6 hours after the end of log phase. And then after the end of the stationary phase, they enter into the death phase, where there is accumulation of a lot of waste materials. The bacteria can no longer metabolize, and then there's more number of cells dying than there are more new cells. dividing. So that's called the death phase. And the other interesting feature that I wanted to mention is about persister cells and then the VBNC condition. So persister cells are, it happens in the nature all the time. So these cells, even though the environmental condition is less than optimal, they essentially become dormant. So there are bacteria like Bacillus subtilis that produce endospore. They produce endospore, and then that's how they survive. But organisms, E. coli, that don't produce endospores, they essentially become dormant. They stop their metabolism, not completely, but almost completely, and they just sit there. And then that's when they become the most resistant to the antibiotics or any sort of disinfectants you're using, because most of the antibiotics are designed to stop some sort of a physiological mechanism, right? Maybe it could be cell wall biosynthesis, or it could be protein or translation. Because now the metabolism is almost stopped, none of the antibiotics are effective because it's just sitting there, but it doesn't die. As soon as there's a favorable environmental condition comes, it just, the persister cell now becomes active and then it starts to divide. So that's something happens usually in the death phase or in the stationary phase. The cell just simply decides to, it senses that there is no more nutrients in the environment. It just basically slows its metabolism down enough that it's not going to die, but it just stays there. And another condition is called the VBNC condition, which is a viable but non-culturable condition. Viable but non-culturable condition, it's interesting because when the cell enters into a VBNC condition, it doesn't divide. It's non-culturable, but it's also not dead at that point. So like another interesting physiological mechanism. It actually happens fairly frequently in the lab, because sometimes I used to store my plates in the fridge for like a week or 10 days. The colonies look really fresh, but then when you re-inoculate, they just don't grow because of whatever reason, they're not able to revive. I guess some of these persister cells, basically, some of them are not able to revive back. So, and that condition is called VBNC, viable but non-culturable. I guess at that point, they're almost equal to being dead. If they're not able to divide, then it's just means they're likely dead. Okay, any question about the growth phase? Sorry, say that again. Selection. Do we talk about selection, like natural selection? Yeah, definitely. Yeah, I mean, I kind of mentioned that, right? It's just hard to really tease apart what's going on, why certain cells are able to survive in the stationary phase, whereas some don't. It just depends on their local environment, I guess. And they're like, when we talk about a strain of E. coli, doesn't matter how much you're trying to keep them uniform, the population has like differences. They're like us, right? We all look the same, but then if you really pay attention, we all look different. Our genetic makeup is different at some level. Same thing as bacteria. I see them as individual organisms. They do have difference in their genetic makeup, and their probably behavior is different. Some of them are able to swim really fast, and others are not. If it's able to swim fast, that means it's going to get all the nutrients. If there's one cell that's not really fed, it's not able to get all the nutrients. So there is definitely a level of natural selection. Natural selection is happening all the time. Yeah. Any other question? Culture systems. So as I mentioned, when the medium is rich, happy environment, they just keep dividing. But in the industry, this is mostly relevant to the industry, probably doesn't happen in nature as much. We're trying to isolate lots of different kind of products, right, that are industrially important, like antibiotics, or we produce a ton of different hormones that are medically important. We produce proteins. We produce alcohol, for example, if you talk about brewing industry, breweries where they produce beer or any other type of alcohol. It's microbiology that's employed in those industries. And you want to, obviously, don't want the bacteria to undergo the stationary or the death phase, because you want the bacteria to produce all these products continuously. And which is why we have these large fermenters, or they're called bioreactors, where the temperature is maintained. There's lots of tubings going on that continuously add the nutrients to maintain a continuous culture, not a batch culture. So in lab, we normally do batch culture. So you have a media, you have a flask with certain media, and then you grow it up to a certain OD or certain turbidity, and then you use it or do whatever with it. But in industries, because you want these products constantly, so they produce these large fermenters, and that's called a continuous culture system. So temperature is maintained, media is continuously added, it's continuously filtered from the other end, because you want to get rid of the waste material too. In fact, the waste is what we were interested in. It could be an antibiotic, or it could be some sort of a product that we're interested in. So that's called a continuous culture system. So here they have given an example of how the antibiotic penicillin is produced, for example. So we have these large fermenters with the penicillium, the mold that produces the antibiotic, and then tons of sugar the mold loves, and then whatever the media, the spent media, is rich in penicillin, and that is what is purified downstream to obtain the antibiotic, and then it is later purified and then bottled and then sold in the market. So lots of issues with the continuous culture system because of such a quantity, and there's lots going on. Often, as I mentioned, the strain changes itself after a certain number of generations. So you have to be careful that it's the same strain. And then there's lots of quality control procedures for that reason. Often they maybe clean out and start over, or there's lots of measures for that. And then contamination is also another issue when you have such volumes or such levels of production of certain things. You want to make sure that it's free of contamination. And again, biofilm is another issue in these fermenters because the tubing, often they get clogged with the biofilms. That's another issue. So we'll talk about more on that later. But yeah, so that's continuous culture system. Any questions about that? Enumeration of cells. This is really important in microbiology, is to have an understanding of how many cells there are. And the term is called the inoculum. Every time you are inoculating a culture for batch culture, continuous culture, it's a good idea to know how many cells you are inoculating. So there's different ways of doing it. There's direct methods and indirect methods. Direct method is essentially directly, you're trying to use microscopy to count how many cells there are. It's not accurate and there's lots of drawbacks, but one of the common method is a hemocytometer assay. Or hemocytometer is usually used for measuring platelets or RBCs, blood counts, but then it's essentially the same thing. It's like a slide that has like a grid. It's got squares that are micron size. I guess it's got millimeter size, and then there is further divided into 0.05 millimeter and 0.25 millimeter, like really small. You'll be able to put that slide, put your culture, your sample. Obviously, you don't want to put your cultured bacteria because you want to dilute it to a certain level, because otherwise your slide will be overwhelmed. So you want to put a little bit of your sample on the hemocytometer, put that slide under the microscope, and then you can basically count it directly how many cells you have. And then based on the volume, you'll be able to calculate back to, let's say you have 5 mils of culture, or your 5 mil of your lake water, for example. You put 100 microliters of that on your hemocytometer, and then you count, and then if 100 microliter has these many cells, how much in 5 mils? You can basically do a simple math, and then you'll get the number of cells. And then, so I had mentioned this, the live dead cell assay. So one of the drawbacks of direct counting is you don't know which ones are dead, which ones are alive. And especially if they're swimming, that's like a whole another problem, because then they'll be swimming all over your slide and then you can't tell which ones you counted, which one you didn't count. So live dead cell assay is actually another neat technique. You kind of get a rough estimate of how many cells are dead, how many are alive. Some of the, and this is based on the principle of the dead cells allow the dye to penetrate, so they look a different color. And then the dyes that don't penetrate, the live cell, have basically an intact membrane. So the dye does not penetrate, so they look a different color. So the live dead cell assay kit you can buy from Invitrogen that allows you to see which ones are alive and dead. But the hemocytometer assay does not allow you to count live and dead cells. It just gives you a rough estimate of the total number of cells in your sample. And then the indirect cell counts, you're measuring the cell counts, it's time consuming, and then it allows you to count the viable number of cells. That's the benefit of doing it indirectly. I don't know, have you done, or do you understand the principle of pour plate and spread plate method? Oh, you guys have done it. Most of you have done it, so I don't have to go through it. So you want to serially dilute your sample, and then you mix it with the medium, and then you pour, and then you incubate, you get a set number of colonies, and then each colony means each cell. That's a benefit. So you can take it for granted that a single, unless it's like a clump of cells that are not separated while you're serially diluting it, but normally a single cell gives to one colony. So depending on how many colonies you have and how much inoculum you used, you can back calculate it and see how many cells you had. And that term is called the CFU, or colony forming units per ml. That's the unit of unit that's often used. And a spread plate method, again, instead of mixing the inoculum with the media and pouring, you simply use a certain volume of the inoculum, and then you spread it using the glass spreader, and then you can count the number of colonies that way too. Serial dilution and plating. Yeah, seems like you already most of you have done it, right? So if you have a culture that's kind of optimal, say in the mid-log phase or in the stationary phase, really thick culture, you want to serially dilute it to be able to bring it to a dilution where counting is easy and convenient. You can probably count up to 100 colonies in a plate at most, but anything more than that kind of gets harder to count, and then you don't get an accurate number. So so serial dilution includes you take 1 ml of the culture and then put it in a tube of 9 ml, then you have 1 to 10 dilution, and then you dilute that, you had 1 to 100 dilution, and so on. You can do as many series as you want. You can even do it in microcentrifuge tubes. If you don't have to do it in these 10 ml tubes, you can have 1 ml of media or water in microcentrifuge tubes, in which case you'll be just transferring 100 microliter from one tube to the next. And then you dilute it 1/10 with each tube, and then you multiply that with the number of colonies you get on the plate, and then that gives you the number of cells. Do you understand what I'm talking about? Anything is confusing?
So you're assuming each colony for a single cell graph?
Yes.
Yeah. There could be cases where there's like 2 cells very close and put together on the plate, right?
Yeah.
Single colony. Just random error we can't count for.
Yeah. So what we used to do in the lab was, after you do the transfer, you mix it really well. You vortex the tube so that even if there's 2 cells that are stuck together, you hope that they separate. So there's not such a high chance that that happens. But at the end of the day, you're not getting a perfect number anyways. These are just close to, and you're talking about 10 to the power 12 cells. There's a little bit of error is normal. Yeah, you're only trying to get a rough estimate. And sorry, go ahead.
You get a lot of overlap.
2 colony, yeah, that happens too. You just use your best guess at that time. Yeah, you can include a standard deviation in your number and say, yeah, whatever. Yeah. And then if you have cells that produce a lot of biofilm or EPS, Rhizobium, for example, it produces tons of EPS, and I was getting a lot of issues with my plate counts to like sometimes huge standard deviations that were not normal, and then realized that it's because they produce tons of EPS. So I have to make sure that it's vortex really, really well. And then I was adding like a detergent, Triton X100. It's like a detergent that helps when there's tons of EPS, it helps the cells to separate so that cells are not as sticky. So there's some tricks that you can use to make sure that the cells are not clumping together and forming a biofilm. If you have lots of clumps, then obviously your numbers are going to be so widely different. So yeah, sometimes detergents, mild detergents, not enough to kill. And then you don't want to acidity dilute in water either, because the water makes the cells to burst, right? So there is so what happens when a bacteria you put it in water? It's going to eventually take up a bunch of water, it's going to swell, and then it's going to burst. So normally you do it in buffer or media that helps the bacteria. Well, media is not ideal either, because you don't want the cells to kind of divide while you're diluting it. Buffer is the best medium to keep the cells happy, not really burst, but at the same time you want to get an accurate number of your plate counts. Any questions related to serial dilution? So yeah, you can serial dilute and do it with via pour plate method or spread plate method. Each have their own drawbacks. Pour plate method, you want to make sure that your media is not too hot. Because while you're pouring, if you are not adding an inoculum, I would put it at like whatever the temperature that I feel comfortable holding my flask with the media. But then if you're doing pour plate with an inoculum in it, then you want to make sure that it is not hotter than say 40 degrees Celsius. So you want to put your media in a water bath and you cool it down, but not cold enough that it solidifies and you get clumps on your plate. So we put it in a water bath that's set at 40 degrees Celsius, and then you add your inoculum, pour your media, mix it, and then incubate. And then the drawback with the spread plate method, I never used to like spread plate method, because when you're using a spreader, there is a bunch of liquid that usually gets stuck to your spreader, and then potentially you're losing a bunch of cells there. So each have their own drawbacks. So there's really no perfect method. So these are just to get a rough estimate of your cell counts. Any questions? No. So if not, that's it for today. And Monday, I will upload a video, so there won't be an in-class lecture. And I will see you on Wednesday. Have a great weekend.
Hello, just a quick
Hello.
question. When is the cutoff date for the midterm?
I'm sorry?
When is the cutoff date for the midterm?
For the midterm, the term test, October 6th, is it?
Yes.
Yeah, it's on your syllabus.
No, I mean, what is the cutoff? Like,
Sorry?
what is the cutoff date?
Current date. Cutoff date. 6th, is it a Monday? What day is it? It's a Wednesday? Oh, okay, so probably up until the Friday the previous week. Sounds reasonable? Yeah.
All right, thank you.
You said written notes are all right for the first test, correct?
Yeah, you can bring your written notes.
If I do my stuff on computer, should I write it as a cheat sheet sort of thing, or?
Yeah, if you want to bring cheat sheet, that's fine, but not on your laptop, but just print it. Yeah.
Like handwritten.
Yeah.
Summarization of things that might be.
Oh, yeah. Yeah, absolutely.
Is there, obviously I'm not going to write everything down, but is there a limit to one sheet or just anything that?
Yeah, anything written is okay. Yeah. Yeah, I mean, you obviously have so much time to go through so many sheets, so you wanna, yeah.